attendance a little bit up today, so some people decide to attend class today. I will wait until the attendance is low, then I'll drop the quiz. So one out of these, four out of the 6 questions will worth 20 points each, and the other 2 will worth 10 points each. 1 out of the 4 questions that is worth 20 points will be a multiple choice. Imagine 1 multiple choice question, 20 points, boom. At least if you don't know anything, you can do eeny, meeny, miny, mo. So, yep, there's a first for everything. Probably this is the first time you'll get 1 multiple choice question that is worth 20 points. Like I told the other group, I can't tell you that the quiz will be hard or it will be easy because those are relative terms. What I can guarantee you it will be is that it will be interesting and entertaining. All right, that is what I can guarantee. Don't be scared. All you have to do is for you guys who attend classes on a regular basis. All right, you take down your little notes, go over the PowerPoint. You have absolutely nothing to worry about. I am wondering if when I drop it, I should have all you guys put on your cameras. What do you think?
No, sir.
Why not?
Sir, because at 8 o'clock I'm still in bed and I haven't, I'm not presentable at that time.
No, but if you had a face-to-face class, you'd have to be up earlier and be presentable before 8 o'clock.
Sir, my room too dark for that, so I just look black, I see me either way, sir.
You just need to put on the light in your room and pull the curtain one
Sir,
side.
we too poor for that, sir. We too poor for that.
Pour for what?
For light, sir, and sun not come out that time at my house, sir.
Where you live? You know, a cave?
Lord, sir.
All right. Folks, sunrise doesn't occur in Roaring Creek until 9 o'clock. While in other parts of the country, sunrise from 5:30, but in Roaring Creek at 9.
9:30 sometimes, sir.
9:30 sometimes, okay. All right, I won't ask you guys to put on your camera. You can still try figure out things and try see how you can. You all might need if you don't have AC in your room, you might need 2 fans to blow on you because you all might sweat a little bit. All right, so let's move on and continue our class. We'll continue, and what we'll discuss right now is we'll go into a little bit more details of the central dogma of molecular biology, right? Central dogma of molecular biology. But before we do that, I'll show you guys this video that has to do with the structure of the DNA molecule.
Structure of nucleic acids, DNA and RNA. There are 2 main types of nucleic acids, DNA, or deoxyribonucleic acid, and RNA, or ribonucleic acid. Nucleic acids are large molecules made up of smaller molecules called nucleotides. The nucleotides in these molecules are linked together through covalent bonds, or bonds where electrons are shared between atoms. Let's start by looking at DNA. The nucleotides that make up DNA have three parts: a phosphate group, a nitrogenous base, and a deoxyribose sugar, or 5-carbon sugar. The phosphate group consists of a phosphate bonded to 4 oxygen atoms, with one of the oxygens bonded to the number 5 carbon of the deoxyribose sugar. An important note here: the number 2 carbon of the deoxyribose sugar is bonded to a hydrogen atom. Carbon atom number 1 of the deoxyribose sugar is bonded to the nitrogenous base. There are 4 types of nitrogenous bases that can be found in DNA: adenine, shown here, guanine, shown here, cytosine, shown here, and thymine, shown here. As you can see, adenine and guanine have double ring structures, and cytosine and thymine have single ring structures. Adenine and guanine are called purine bases, and cytosine and thymine are called pyrimidine bases. DNA is a double-stranded nucleic acid, and its molecules take on a helical formation. Each helical chain has its phosphate sugar group toward the outside, and the nitrogenous bases facing inwards towards the nitrogenous bases of the other chain. Each of the bases on one chain is joined to the base of the other chain through either 2 or 3 hydrogen bonds. Thymine and adenine are joined by 2 hydrogen bonds, and cytosine and guanine are joined by 3 hydrogen bonds. Thymine and adenine are always a base pair, and cytosine and guanine are always a base pair. It's estimated that DNA molecules contain more than 100 million of these base pairings. And in one individual's body, the sequence of the base pairings is the same in every DNA molecule. So this sequence of base pairing is unique to that individual. DNA is often called the information molecule because it contains the master code needed to make various RNA molecules and protein molecules in the body. Now for RNA. The nucleotides that make up RNA are very similar to those that make up DNA. RNA nucleotides have a phosphate group with the same structure as in DNA. They have a 5-carbon sugar, but the number 2 carbon is bonded to a hydroxyl OH group instead of a lone hydrogen atom. And this sugar is called ribose. It also consists of the 3 same nitrogenous bases as DNA: cytosine, adenine, and guanine. But it does not contain thymine. Instead, it contains uracil, which is a single ring structure like thymine, making it a pyrimidine base. Most RNA molecules are single-stranded nucleic acids, and many times they form a folded, compacted structure with some hydrogen bonding taking place within the base pairs of the molecule. These pairings are the same as in DNA. Cytosine and guanine are a base pair, and uracil takes the place of thymine and pairs with adenine. RNA is extremely important in the process of forming different proteins in the body, of which we will cover in depth in a later video. And that be the basics on the structure of DNA and RNA.
Okay, so what you see on your screen here is the central dogma of molecular biology, which you guys as biology students, you should know like in the palm of your hands, whereby you have a DNA molecule, and every time mitosis is taking place, we have cell division, DNA must replicate to pass copies to the progeny cells. Okay, so you have DNA replication. And then you have transcription taking place, whereby in the case of replication, the entire DNA molecule replicates. It makes a copy of itself and sends to the progeny cells. But in the case of transcription, it is not the entire DNA molecule that is transcribed. Only portions of that DNA molecule is transcribed. And each portion that is transcribed and produces a messenger RNA, that portion is called a gene. Okay, so remember that there are portions of that DNA molecule where transcription does not happen, And so those areas on that DNA molecule actually don't code for anything. Okay? So in transcription, you'll end up having a messenger RNA molecule that is taking information from the DNA molecule over to the ribosome, where translation will take place, all right? And that is the production of proteins. Okay, so that right there is the central dogma of molecular biology that you guys need to know very well. In DNA replication, the enzyme that plays a key role and catalyze that reaction is the DNA polymerase. All right, so the DNA polymerase is responsible to make additional copies. So it will cause DNA to denature, exposing the bases, and then that enzyme is responsible to bring the corresponding bases to build a complementary strand on both template strands. And then you end up with 2 DNA strands, right? 2 DNA molecules. In the case of transcription, whereby you have that RNA molecule being produced, the enzyme that catalyzes that reaction is the RNA polymerase. All right, notice here that you end up with a single-stranded RNA molecule. Why do you end up with a single-stranded RNA molecule after transcription? When you have a DNA molecule like this, which is double-stranded, during transcription, portion of that molecule becomes denatured, and transcription only uses 1 strand, which is the template strand, to build that messenger RNA. So this strand, it uses this strand as template to build that messenger RNA. The other strand, the strand at the top, is called a non-template strand. All right, so we'll get into this in more details when we discuss transcription. But when this messenger RNA is built, then that messenger RNA comes out, all right, and then it goes to the ribosome. It's a single-stranded molecule because it was built from only the template strand of that DNA molecule. And of course, when transcription is done, then this DNA molecule renatures, it comes back together, all right? And here you have translation, which takes place in the ribosome, all right, whereby you have a protein molecule is being built, all right? So that completes this whole process of the central dogma of molecular biology, from DNA to proteins. Bear in mind that all enzymes play a key role in our bodies, in the bodies of all organisms. So each enzyme is protein by nature, and so each enzyme had a gene on that DNA molecule which is responsible for its production. If I can recall, in a previous class, I said that albinism, all right? Albinism is a case whereby it results from a defective gene that is responsible to produce an enzyme called tyrosinase. So let's assume this is the gene right here that is responsible to produce that enzyme tyrosinase. So this gene has a defect. And because that gene has a defect, it cannot produce the tyrosinase that plays a key role in the production of melanin, which is the pigment in your skin, all right? So just imagine a gene that is responsible to producing an enzyme being defective and can lead to something like albinism, all right? But let's get into each one of these steps a little closer. We go in a little bit more detail. So we'll discuss DNA replication, all right? So DNA replication in prokaryotes. So remember we said that prokaryotes has 1 single circular DNA molecule, that chromosome, right? It says DNA replication in E. coli and other prokaryotes occurs by a semi-conservative mechanism. in which the strands of a DNA double helix separate and a new complementary strand of DNA is synthesized on each of the two parental template strands. Take note of that word, those words you see there in bold, semi-conservative mechanism. I'll explain that in a little bit more details, okay? By the way, remember that what we're discussing here will not be on the upcoming quiz, right? This will be on test number 2. Semi-conservative replication results in 2 double-stranded DNA molecules, each of which has 1 strand from the parent molecule and 1 newly synthesized strand. So if we look at this diagram here, this is basically what semi-conservative DNA replication is all about. At the top here, the top right, here we have the original DNA helix. What takes place after that for DNA replication? You must have denaturation, which means that that double-stranded DNA molecule must open up and separate. So when it open up and separate, for example, all right, I'll use my own diagram over here. So this is the DNA molecule that has been denatured. So after it has been denatured, then you have the complementary strand being built on those exposed bases. And remember we said that it is the DNA polymerase that is responsible to bring the corresponding bases to build that complementary strand. All right. So what you see here at the bottom right is the DNA helices after 1 round of replication. So take note that each molecule has 1 strand from the parent molecule or the original molecule, which is what the one you see in blue. Hopefully nobody is colorblind here and can discern which one is blue. And the one is in yellow, all right, would be the newly synthesized strand. So after 1 round of replication, you would end up with these 2 molecules, and each one of them have 1 strand each of the original DNA molecule, and the other strand would be the newly synthesized strand. Folks, that is what's because it conserves. One strand from the original DNA molecule, okay? It was proposed some time ago, several models were proposed of how DNA actually replicates. So the first one was conservative. And this model indicates that you have the original DNA molecule here, and that molecule, the entire molecule was used as a template to build a new DNA molecule. That was the conservative model. It conserves the original DNA molecule. The one I explained a while ago, which is the semi-conservative, in that particular case, it conserves the 2 strands from the DNA molecule, and then it builds a complementary strand to each of these molecules. So you end up with 2 molecules that have 1 strand from the original DNA molecule and the other strand being the newly synthesized strand. And then the other model that was proposed back then was the dispersive model. And in this particular model, you have the original DNA molecule and you have portions, all right, maintaining that original DNA molecule and other portions being newly synthesized and so on. We now know that DNA does not replicate using the conservative nor the dispersive model, but rather using the semi-conservative model. Okay, so that is how DNA replicates. Okay. DNA synthesis enzymes. Four components are needed for the in vitro synthesis of DNA. What does this mean? When you hear in vitro synthesis of DNA, what do you think we're talking about? What does in vitro means? Anybody knows? In vitro synthesis of DNA. Synthesis means to make, right? So it means that we're going to make DNA somewhere. What does in vitro synthesis of DNA means? Anybody? You can do like you would normally do during a test or a quiz. Consult Uncle Google, because that's what you normally do. You're used to that. In vitro refers to inside the lab, all right? Inside the lab, like what Kaylee says, it's done in the lab. So when we talk about in vitro synthesis of DNA, we're not talking about DNA replication taking place in your bodies right now. We're talking about actually getting a DNA molecule, take that to the lab, and you can make more copies of it. So in vitro synthesis of DNA is nothing more than referring to what PCR does, polymerase chain reaction. I'm sure you've heard of PCR, which stands for polymerase chain reaction, right? So basically what that does is to make more copies of a DNA sample that you have, and that is done in the lab. So there is this machine, the thermal cycler, that can actually do that. It mimics DNA replication. But for you to be able to do that, several things are needed. These things include nitrogenous bases. So all four deoxyribonucleoside 5' triphosphates, which would be the DATP, DGTP, the DTTP, and DCTP, which are collectively called DNTPs. All of that is needed, because remember, you're going to build and make more copies of that DNA sample that you have. So you need these nitrogenous bases to build a complementary strand. So it says if any one of these out of the four is missing, synthesis will not occur. Because remember that a sequence of DNA would be like, for example, A, T, G, C, and so on, right? And so if the corresponding base would be a thymine, but there's no thymine, there is no DTTP, then the reaction would stop right there. Okay? So all four of these DNTPs need to be present for in vitro synthesis of DNA. Magnesium ions in the form of magnesium chloride plays a key role as a cofactor. All right, a cofactor is something that is needed to be there for an enzyme to be able to function. All right. A fragment of DNA to act as template. So you need a sample of DNA that you want to make more copies of. So for example, if we're talking about a crime scene, you go to a crime scene and you get a piece of hair. Well, the DNA in that hair will be that fragment that will act as template, because the DNA from that piece of hair, that is the one that you're going to make more copies of. Okay? And of course, you need that enzyme that will catalyze the reaction, which is the DNA polymerase. And in this particular case, they use a special DNA polymerase, which is called TAQ polymerase. All right, TAQ, TAQ polymerase. They use this one because you all know that what happens to proteins and enzymes at high temperatures? What happens to that? They become denatured, right? They're denatured. So in the in vitro synthesis of DNA, remember I had explained when Shian asked the question earlier, how is it that you can actually split a DNA molecule? And I said with what? Heat, high temperatures. So we need high temperatures here to denature the DNA molecule, split it in single strands, but at the same time, we can't use an enzyme that will become denatured because of that heat. And so a Taq polymerase is a special enzyme that can withstand high temperatures. All right, so the Taq polymerase is used because it can withstand the high temperature required to denature that DNA molecule. Okay. Let's look at the molecular details of DNA replication. What takes place during DNA replication? The initiation of DNA replication in prokaryotes requires the local denaturation of the DNA molecule at a specific DNA sequence, which is called the origin of replication. So remember that whenever we talk about denaturation of DNA molecule is to break those hydrogen bonds and move it from double-stranded to single-stranded, right? That is what denaturation of the DNA means. So what it is saying is that in the case of prokaryotes, remember prokaryotes have one single circular DNA molecule. So what it is saying is that, and this is double-stranded, so let me see if I can, all right? Double-stranded. So what it says is that at a certain point on this molecule, it is called the origin of replication, and it is right from here where denaturation will take place. So this will open up, and that is called the origin of replication. So denaturation will start from there, and it will start to open up, where complementary strands will start to build in both directions. The origin of replication is that point on the DNA molecule where denaturation starts and replication actually proceeds from there. So at that site, the double helix denatures into single strands, exposing the bases for the synthesis of new strands. So in a circular chromosome, such as is found in E. coli, the local denaturing of the DNA produces what is called a replication bubble. All right? So if we look at this diagram right here, imagine this to be the origin of replication on that circular DNA molecule. Right where you have denaturation taking place, where the DNA opens up into single strands, that is called the replication bubble. So what you see here, right here, is the replication bubble. And then this portion here, right here, is called a replication fork. And this right here is another replication fork. Replication will proceed in both directions. So this will be occurring in this direction, and this will be also occurring in this direction, okay? Until the entire circular molecule has been replicated, okay? A little bit down the road, I'll explain about leading strand and lagging strand, okay? So here you have the circular DNA molecule. This is the origin of replication right here. Notice that it has been denatured, and right there you have the complementary strand being built already. So this right here will be the complementary strand on that top strand, and then this would be the complementary strand for the bottom portion. All right, so this would proceed. Remember, this is the replication fork. and this will proceed in both directions until it reaches the terminus, where you'll end up having two circular DNA molecules. All right. In E. coli, there is a single origin of replication, which is called the ORIC. All right. So that origin of replication is called the ORIC, from which replication proceeds bidirectionally in two directions. An initiator protein binds to the parental DNA molecule at the origin of replication sequence. All right, so an initiator protein binds to the parental DNA molecule at the origin of replication sequence. So if we look at this diagram here, it says bacterial chromosomes have a single point of origin. So a single origin of replication that is observed right there. So if you notice, that is the replication bubble. And what is taking place? You can notice that the complementary strand that you see in red is being built on both strands. And so we said that replication proceeds in both directions. All right. If you notice here, we talk about the semi-conservative mechanism of DNA replication. It is observed right here in this diagram, whereby you see the two strands from the old DNA, which is in gray, and the newly synthesized strand, which is in red. All right? And so this will continue until you end up having these two molecules of DNA. Okay? The two DNA strands then separate in that region. This involves a localized denaturation, which creates the replication bubble, and that requires the physical untwisting of the DNA. This untwisting, which uses energy derived from the hydrolysis of ATP, is catalyzed by the enzyme DNA helicase. All right. So now we have another enzyme playing a role in DNA replication, which is DNA helicase. So what is the role of DNA helicase? It helps to denature the DNA molecule. All right. So DNA helicase helps to denature the DNA molecule, and after you have single-stranded DNA molecules, then you have the DNA polymerase that will come and bring the corresponding bases to build that complementary strand. Okay? So take note of all the enzymes that play a key role in DNA replication. Here we have another enzyme, which is called DNA primase. The DNA primase binds to the helicase and the denatured DNA. And the role of the primase is it will create a short primer, which will anneal to the DNA molecule from where polymerization will start. All right? So primase is derived from RNA polymerase. So basically what it does is that it will create a small portion of, which is called a primer. All right, so if we look at this and you have these molecules here, so the primase will build a primer, and that primer will attach itself right here, for example, and right here. And it is from where that primer anneals, there is where polymerization will start. So this will continue to build in this direction. And this one will build in this direction until both strands have completed their complementary bases. All right? So that is the role of the primase. All right, so you have DNA polymerase, DNA helicase, and DNA primase. These are three very important enzymes that play a key role in DNA replication. All right, so folks, we will stop there for today, and on Tuesday, we will discuss this diagram. So this diagram will explain the role of all these enzymes in DNA replication. Okay, any questions before we leave? All right, if there are no questions, be reminded, guys, that if you are free around 10 and you're on campus, remember that there's a fundraising event for ex-UB student who is in need of some medical attention. I sent you guys the flyer, so if you can make it, all right, please make sure that you reach out.